Hello class, Mr. Allman here, and this is your Allmancast follow-up additional HL material on the topic of respiration. Before we get into the details of the processes involved in each individual step of aerobic respiration in animals, let's take a step back and consider a little bit of cell biology. Now, you should remember that energy cannot be created or destroyed. All cells and all living things need energy as a way to be able to do work. There's a lot of work involved in staying alive. Energy is stored in chemical bonds, and so respiration is about being able to release that stored chemical energy to be able to produce as great an amount of ATP as possible. And with our study of photosynthesis, we saw that the chloroplast was a uniquely adapted organelle within cells that was responsible for carrying out the capture of solar energy into ATP and carbohydrates. Now we're going to look at the mitochondria as a uniquely adapted organelle, which is structured to be able to release the stored energy in nutrients like glucose. In considering the structure of the mitochondria, you should link this back to the topic of endosymbiosis, how mitochondria used to be free-living prokaryotic organisms that were really good at being able to extract a lot of stored energy from nutrients. Once these organelles were taken in and became a part of these larger eukaryotic cells, they continued that function. Being fed nutrients by the larger cell, they would break them down and produce that ATP to share with the larger cell. The structure of mitochondria consists of two membranes. An outer membrane, which gives the mitochondria its characteristic pill shape, and a highly folded inner membrane. More folds are going to increase, remember, surface area, which is going to be very important for the process of phosphorylation or ATP production, which I'll cover a little bit later on in this video. The folds have a more specific name called cristae. So when you look at an image, a cross-section of a mitochondria taken through an electron microscope, you can see those folds as they extend across the length of the organelle. Also within mitochondria, there are free ribosomes. Free ribosomes, remember, will produce proteins, and many of those proteins are enzymes that are involved in some part of the respiration process. There's also mitochondrial DNA, and that DNA contains the genes necessary for those ribosomes to be able to actually translate those needed enzymes. As we look at the details of the biochemical processes happening in the breakdown of nutrients during respiration, there's a couple of terms that are going to come up consistently that you should be familiar with. Oxidation is a type of chemical reaction in which a molecule, here symbolized by A, is losing electrons as part of that chemical change. Those electrons, which are symbolized here with those little red E's, those negatively charged electrons are going to be lost. Therefore, as a consequence, we're going to see an increase in charge, because if molecule A has 2 less electrons, it's going to have more positive charge at the end of this process than it did at the beginning. The opposite reaction is called a reduction. In a reduction reaction, molecule B here is going to be gaining electrons. So as a product of the reduction, it's going to be reduced as it increases in its number of electrons. And so depending on what else is going on, this may cause a decrease in charge, because now molecule B is more negatively charged with the addition of those electrons than it was in the beginning of the reaction. As you can see, in many cases, an oxidation and reduction reaction are coupled, meaning that one molecule is oxidized and those electrons are immediately transferred over onto a second molecule, which is simultaneously being reduced. A helpful acronym that you can use is oil rig. Oxidation is losing electrons, while reduction is the gaining of electrons. So please try to apply these terms in your study of respiration. Again, looking at the process of respiration as a whole, we'll begin in the cytoplasm, where glycolysis is occurring. and then we'll move into the mitochondria for the next steps where most of the ATP production is going to occur. We'll begin with glycolysis, where we will have substrate level phosphorylation, meaning we're going to be phosphorylating some ATP. Then during the Krebs cycle, we're going to create some more ATP within the mitochondrial matrix, but the majority of ATP production is going to be through the process called oxidative phosphorylation, where it's coupled with chemiosmosis to be able to produce large amounts of ATP. As a review of glycolysis, we're starting with a nutrient like glucose. We're dividing it into 2 molecules of pyruvate. Each of those molecules of pyruvate will have 3 carbons. This is actually a multi-step process to get from glucose to pyruvate, and I'll show you the details of that on the next slide. We're going to require 2 ATP in an energy input phase, but 4 ATP will be produced, so we have a net production of 2 ATP for each glucose molecule that we start with. So glucose will be oxidized, meaning that electrons are going to be lost from glucose, and those electrons will be transferred over to a molecule called NAD+. So NAD+ will be reduced while glucose is simultaneously being oxidized. Here you can see the full pathway. There are really 10 steps to glycolysis, beginning with glucose here at the top. You can notice above the arrows here, you can see in red the names of the specific enzymes that are associated with the processes, and also in places where ATP is either used or consumed. And so to go from glucose here at the top to pyruvate here at the very end, ATP is going to be used a couple of times. ATP will be used once to be able to make a more highly energized form of glucose, which will allow it to become slightly less stable and therefore acted upon by future enzymes. And another ATP is utilized in the splitting of a 6-carbon molecule down into 2 3-carbon molecules. The rearrangement of those 3-carbon molecules into pyruvate is actually where ATP is then going to be generated at a few different steps. So the ATP use and ATP harvest is actually coming across really in different stages of this 10-part chemical reaction. There's also going to be oxidation, and so you can see here in blue the transfer of NAD plus into NADH, which is happening twice because each of the 2 3-carbon molecules will go through the processes here underneath this blue arrow. This is the part where electrons are actually being removed from the nutrient. Those electrons are being removed as the nutrient is oxidized, allowing for the reduction of NAD into NADH. NADH is therefore what we term an electron acceptor. It is taking those electrons by being reduced, and it's going to transfer those electrons on to a future step of this process. Again, to summarize, we have an energy investment phase in which glucose is going to be split, ATP is utilized. Then in the energy payoff phase, we are creating ATP from the rearrangement of those 3-carbon molecules, and we are also oxidizing those 3-carbon molecules, allowing us to be able to produce reduced NADH, which again will be transferred to a future step. The ATP can be utilized immediately as a source of energy within the cell. Throughout these processes, please pay attention to both the inputs and outputs of the process, because that is a helpful way to be able to summarize what it is that's actually happening in each of these stages of respiration. With glycolysis done, now the products of glycolysis, the pyruvate and the NADH, are going to move into the mitochondria. And so now we're going to look at what those steps are as we continue with the aerobic portion of respiration, when oxygen is going to be utilized in the further breakdown of pyruvate. So we have the pyruvate and the NADH entering into. We're going to look at in detail the link reaction, the Krebs cycle, and the electron transport chain as it works to be able to power the production of large amounts of ATP. So first, the link reaction, which, as the name suggests, is going to link together the outputs of glycolysis with the beginning of aerobic respiration. we're going to have a decarboxylation of pyruvate. And if you break down that term, to decarboxylate, we are taking away carbon from the 3-carbon pyruvate molecules. So we are taking away carbon. It's going to form a molecule of CO2, which will be released as a waste product. Pyruvate is also going to be oxidized again. So you can see here at part 2, NAD is going to be reduced into NADH because electrons are being taken from that pyruvate. And then what's left over from the pyruvate after carbon dioxide and the electrons are removed is going to merge together with a very large molecule called coenzyme A, or CoA for short, to form a molecule called acetyl-CoA. So that acetyl-CoA is then going to be the molecule which will enter into the next stage of the process of respiration. So again, to summarize there, we start with pyruvate and the reducing agent NAD+. Then the outputs of that are going to be acetyl-CoA, which is a 2-carbon. The acetyl is the 2-carbon left over from pyruvate, where it combined with coenzyme A. We get a waste product of 1 molecule of carbon dioxide, and then we get a reduced NADH. Each of that is going to happen 2 times per glucose, and that is going to carry over into all the future steps, because remember, we have 2 pyruvates as the product of glycolysis. From the link reaction, we'll move on to the Krebs cycle. The Krebs cycle has a synonym called the citric acid cycle, because citric acid is one of the first components of this cycle. As the name suggests, the Krebs cycle is cyclical because there's a series of reactions in which we get a starting product, which is going to break down into an ending product, which will then get recombined to form the starting product again and again and again, with the input of additional acetyl-CoAs each time. In this process, we are combining that acetyl-CoA with a pre-existing 4-carbon containing molecule, which is the leftover at the end of a turn of the Krebs cycle. That combined larger 6-carbon molecule will then be oxidized, meaning that more electrons are being removed from the nutrient, and it will be decarboxylated, and the decarboxylation will remove carbon, with the whole goal here of being able to produce greater amounts of NADH and a second electron acceptor, which has the same function, though it's a different chemical structure, called FADH2. And together, those electron accepting molecules are going to move on to the next stage. We're also going to make a small amount of ATP, and we'll produce carbon dioxide as a byproduct. Looking at the process here, we begin at the top. You can see the link reaction is actually going to be summarized here in this image. You can see pyruvate, which is broken down, decarboxylated and oxidized into acetyl-CoA. That acetyl-CoA is then going to merge together with the ending molecule, oxaloacetate, which is a 4-carbon containing molecule summarized here on the left. The coenzyme A is actually removed and reused in the link reaction again. And we make now a 6-carbon containing molecule called citrate, or often citric acid. Then we're going to go through a series of reactions. Again, we have in red the names of the specific specific enzymes that are utilized as part of this process. We are reducing it. So again, in blue here, you can see NAD+ being reduced into NADH. That means that the carbon-containing molecule has been oxidized as electrons have been stripped away. That oxidation of the nutrient is happening again. And also you can see 2 carbon dioxide molecules happening back to back in this part. As we went from a 6-carbon containing molecule here in the brown to a 5-carbon containing molecule with the reduction of a CO2 in green. And then now we're at a 4-carbon containing molecule because another carbon dioxide was lost as this nutrient was decarboxylated. Here you can see at the bottom we have the production of ATP. And then here we have another carbon-containing molecule, FADH, into FADH2. Here the Q is actually used as an alternate abbreviation in this image for FAD. and we have another reduction of some more NADH. So you can see at many different parts here, we are oxidizing the nutrients, releasing electrons. Those electrons are being taken by these electron carriers, and they're going to be used again in our next step. In terms of numbers here, we start with an acetyl-CoA for every turn of the Krebs cycle. We start with 3 NAD+, 1 FAD, and 1 ADP. And then as we decarboxylate, oxidize, and remove energy from those nutrients, we're going to get carbon dioxide as byproducts. And then our intended products are the NADH, 3 of the electron carriers, 1 of the FADH2s, and then an ATP. Again, all of that being repeated for each 2 times for each glucose. All right, at this point, we've only made a little bit more ATP, so we still haven't accomplished our goal, which is being able to produce large amounts of ATP from those nutrients. So now we're going to get to oxidative phosphorylation, which is a paired series of processes, the electron transport chain and chemiosmosis, which work together to be able to produce that ATP using predominantly those electrons which were being harvested from the nutrients in the previous stages. In this process, what we're doing is that we are oxidizing NADH and FADH2. So again, this is the opposite process to what was happening earlier. Before we were creating these by reduction reactions, now the electrons themselves are going to be removed from these temporary electron carriers, and those electrons are now going to be passed between a series of bound integral membrane proteins, which are located on the inner mitochondrial membrane. So this is referred to as the electron transport chain, because electrons from those electron transport carriers, the movement of those electrons is being symbolized here by this yellow arrow, being passed from one protein to the next in a chain, eventually combining together with oxygen gas and protons to be able to produce water, H2O, which is one of the byproducts here of this process. Now we finally see the role of oxygen as part of aerobic respiration. Oxygen is the thing that actually accepts the electrons in the end. The movement of those electrons is useful in being able to produce the actual ATP, but those electrons need to go somewhere. They can't be left to move around freely forever. They always need to be bound to other molecules in order to control the energy that they have. And so ultimately those electrons, once their usefulness is over, will be combined with oxygen to be able to make water. If oxygen is not available, then there is no place to put those electrons, and that's what halts all of these processes. Now, during the process of the passing of those electrons from one protein to the next, these integral membrane proteins are actually protein complexes, multiple proteins combined together. And what they do is that they use the energy from the passing of those electrons to power an ion pump. Remember, ion pumps use energy to be able to move materials from where they are less highly concentrated towards where they are more highly concentrated against the direction of diffusion. So what's happening here is that the energy from the electron is pumping protons, H plus protons, on the outside into the intermembrane space of the mitochondria. Again, this is the inner membrane of the mitochondria. Having a folded inner mitochondrial membrane increases the surface area, which allows more space for these protein complexes to do their work. We are creating a proton gradient. The proton gradient means we are creating a high concentration of protons here on the exterior part of this inner mitochondrial membrane. This is called the intermembrane space. Because the intermembrane space is very small in volume, it is much quicker and easier to be able to create that proton gradient. So the electron transport chain here is this first section here on the left, electrons being passed, protons pumping into the intermembrane space, water being produced as oxygen is going to be reduced with those leftover electrons. Now we'll go on to the second part here, chemiosmosis, which is where we're actually going to produce these large amounts of ATP. This is our big finish. ATP production is going to happen through an important membrane known as ATP, an important membrane-bound enzyme called ATP synthase. It is synthesizing ATP. The way that it does that is by utilizing the proton concentration gradient that was formed from the electron transport chain. When you create a gradient of protons, facilitated diffusion is going to allow that proton gradient to move those protons from the intermembrane space where they're highly concentrated back into the mitochondrial matrix where they are less highly concentrated simply by diffusion. That process does not require energy. However, the movement of those protons through ATP synthase is going to utilize that kinetic energy to be able to synthesize ATP by phosphorylating molecules of ADP with free phosphates, which are going to be found located in the mitochondrial matrix. As we look more closely at this, you can see again the protons, which are highly concentrated on the exterior. Those protons are going to move through a special proton channel found in the membrane-bound portion of ATP synthase. Those are going to cause this membrane-bound part to rotate. And the rotation of that moves this arm, called the stator of the ATP synthase, which then exposes the active site on the catalytic knob of ATP synthase. So the movement of the protons opens up the active site, causing adenosine diphosphate and a free inorganic phosphate to combine in the active site, and then the enzyme will catalyze the production of ATP, the product, and that product will then be released. Then the active site will then be exposed so that additional protons will be able to, again, turn the ATP synthase, allowing more ADP to be converted into ATP. As long as you have a supply of protons, then ATP will continue to be produced. In oxidative phosphorylation, the electron transport chain is going to utilize the electrons which are bound to our electron carriers, NADH and FADH2. We also need oxygen gas as an electron acceptor. And as the outputs of this, we oxidize those electron carriers, making NAD plus and FAD plus, which are going to return back to back to glycolysis, the link reaction, and the Krebs cycle, where they can be reused and re-reduced. We're going to produce water, which is a byproduct, which will either remain in the organelle or exit back into the cytoplasm. And we're going to produce a large quantity of ATP. There's not a specific amount because really these processes are decoupled from the breakdown of glucose, but something in the mid-30s is a good estimate for the amount of ATP that we can produce, giving us on the whole, something up to 36 to 40 ATP being produced on average per glucose molecule between the combined processes of glycolysis, Krebs cycle, and oxidative phosphorylation. So that summarizes the biochemical details of what's happening to nutrients as electrons are being removed and they are being broken down, and that energy is being utilized by ATP synthase to make large amounts of ATP. All right, there's a lot there. Please let me know if you have any questions. Certainly go back and check again for any of those steps that you might have missed the first time. Thanks for watching.